This is the second half of chapter 21. We were talking about genome evolution. And in the first half, we discussed the fact that genome sequences are constantly changing due to mutations that are altering gene sequences. And as we've discussed in some previous chapters, we know these changes are helping drive the evolution of physical changes in organisms. And if we look just at the genome level, we're seeing these phenomenon of lateral gene transfer and gene duplication, causing genomes to grow and causing the production of new versions of genes over time. But what I wanted to point out here is that it's become pretty clear as we've studied genome evolution that the genome sequences may not actually even need to change in many instances in order to get um, radically new morphology, new body forms. We could simply alter the expression patterns of genes without changing their sequences at all, and that can cause these new body patterns to arise. And so this has probably been best and was really first studied in the fruit fly Drosophila, which is, you may or may not be aware, they've got segmented bodies, and we see these segments appear very early in some of the larval stages. I'm gonna show you some images here in a second. Um, and even at these early stages of embryonic development, we know that the fates of these cells have already been determined. And this is really referring to the fact that in embryos, really of any multicellular organism, you begin with what are called stem cells, embryonic stem cells, that are very generic cells that have the potential to become any specialized cell type in the adult body. Um, and usually these embryonic stem cells will go through this process of determination and then differentiation, and they take on these specialized fates. Um, and in studying fruit fly development, became clear that these fates were determined in most cases pretty early on, even at some of these early larval stages. Um, but the reason I'm talking about fruit fly larval stages is because I wanted to give you this example of gene expression altering morphology in fruit flies, and this is referring specifically to what are called the Hox genes. Okay? And so the Hox genes are genes that encode transcription factor proteins, and you may remember from previous chapters, transcription factors are these proteins that will alter the transcription of other genes, right? They'll bind to promoters and help RNA polymerase initiate transcription of certain genes. And in studying the fruit fly genome, we see this is a gene family, similar to what we talked about with hemoglobin in the last section. So there's a bunch of these different Hox genes. You can see them shown here in the fruit fly genome, sort of color coded. Each one of these colored bars would be a different Hox gene. Um, and here's an example of that segmented fruit fly embryo, right, larval stage. Um, and it turns out that specific Hox genes are expressed in very specific parts of the embryo. So, for example, this orange Hox gene here is just being expressed. The protein coded for by this gene is just being produced in this little cluster of cells up here at one end of the embryo. And likewise, this other Hox gene here, represented by the green stripe, is only being expressed in these cells back here at the other end of the embryo, and so on. And what you can see at the top here is that these different sort of color-coded sections of the embryo are going to give rise to these different portions of the adult fly. Okay, and so it turns out that where these individual Hox genes are expressed is actually what's determining this overall morphology and body pattern in the adult fly. And these Hox genes are not just found in fruit flies, so we find them in pretty much all animals, including humans. But here, just for the sake of illustration, you can see there's a set of Hox genes in the mouse genome and that are expressed in the mouse embryo in a very similar pattern to what we see in fruit flies. And there's a almost identical set to that found in mice in the human embryo as well. So this is a sort of a classic example of homologous genes, right? We're seeing these very similar genes shared across the genomes of multiple organisms, presumably due to some common evolutionary history, some shared ancestor that also had those particular Hox genes. But coming back to this basic idea of just changing gene expression to alter morphology and drive evolution, um, the importance of these Hox genes really came to researchers' attention um, through the discovery of what was called this antennapedia mutation, where they found fruit flies, which normally have antennae, right, growing out of the front of their face, as you can see here. Um, they found these mutants where antenna had been replaced by legs. So these were not just, strangely shaped antenna. These were like fully formed legs growing where the antenna should be. And as researchers dug into this, they realized what had happened is just a slight change in the expression pattern of specific Hox genes, and these came to be known as homeotic mutations, and they've since found lots of different homeotic mutations, so change in Hox gene expression pattern. But just to go back, right, so, um, you know, this means basically all that happened was the Hox gene that, let me go back one more, that basically is going to direct the formation of the leg in the adult fly. So let's say the orange one here, right, that normally gets turned on in this portion of the embryo. This particular Hox gene accidentally got turned on up here in some cells, a little cluster of cells at the front of the embryo. So instead of producing antenna, we produced a leg, right, where it shouldn't be. So what this illustrates is just by tweaking the expression pattern of these Hox genes in an embryo, whether you're talking fruit flies, mice, humans, right, pretty much any animal, we can sort of rearrange body pattern and get these dramatic new body shapes without even having to, create new genes or even alter gene sequences at all. And so while we're talking about changes in gene expression, I wanted to point out that there's really three main ways gene expression patterns can change. So there's what's called uh, heterometry, which is differences in the amount of gene expression. And sort of the classic example of this is the beak sizes in the Galapagos finches that were studied by Darwin. It turns out on the molecular level, the beak size is regulated just by the amount of specific proteins that are produced by two key genes. And if you have more of those proteins being produced, you're going to get thicker, fatter beak. If you have less, you're going to get a thinner, pointier beak. Okay? So that's heterometry, just changing the amount of that a particular gene or a handful of genes are being expressed. Then you have what's called heterochrony, which is changes in the timing of gene expression. So the amounts don't necessarily differ, but the length of time, or maybe the time during development at which these genes are being expressed, is altered. And the example I want to show you here is the difference in neck length between giraffes and humans. And if we look anatomically, the skeletal structure is essentially the same. We have basically like the same number of vertebrae in the neck between giraffes and humans. Of course, the giraffe's neck is much longer because, as we can see here, the vertebrae themselves are much longer, right? In the human neck, we've got these, very thin vertebrae compared to these long bones in the giraffe neck. And it turns out this difference just comes down to the timing of one specific gene that basically stops bone growth from occurring, right? So um, that gene gets expressed earlier in human development, so bone growth is stopped earlier than it is in giraffes. But the overall process is exactly the same. The genes that are involved are basically the same. It's just this difference in timing, and that would be called heterochrony. And then the last is heterotopy, which is differences in the spatial expression pattern of genes. So, like which tissues or which cells the genes are expressed in. Um, and the example I wanted to give you here is the difference in webbing between the toes of chickens and ducks. Obviously, ducks have webbing between their toes and chickens do not, right? As you can see down here. Um, and it turns out during embryonic development, both actually have webbing between their toes. It's just in the chicken toe, a certain gene gets expressed that will cause that tissue to carry out apoptosis and go away. And even though ducks have that same gene, it is not getting expressed in the same tissues. So we have that one difference in the the tissue specific gene expression pattern that's giving you this difference in morphology. So basically, what I hope you're walking away from this with is yes, we can change the sequence of genes, and of course, that can alter gene function and morphology. But really, just as powerful, we can just change the expression patterns of genes in various ways, and that can also shape evolution in a very dramatic way and create new body forms. And you put those two things together, right? And maybe it's now not surprising that evolution is constantly producing new morphologies and new body forms. So to finish this chapter out, I just wanted to touch on, well, what are some of the applications of studying molecular evolution? And really from a biological standpoint, we can use an understanding of gene evolution to um, get a better understanding of how specific genes and proteins work. And typically, this would be done by doing sequence alignments like we talked about in the first half, and producing what are called gene trees. So these would be like phylogenetic trees that are just comparing the sequence of maybe one specific gene across a bunch of different organisms. What you're seeing here is a gene tree for a gene called the engrailed gene, which makes this protein that's very important for development in pretty much all multicellular organisms. Um, and by understanding the evolutionary relationship of a single gene across these different species or within one gene family, right, we can really uncover some of the secrets to how this gene functions, and how it may be affecting organismal biology as a whole. So that's sort of from the academic standpoint, right? Our understanding of biology and how life works can really be improved through studying molecular evolution. But in a more practical sense, there's all kinds of things you can do. Maybe one of the more pertinent right now, where we're in the middle of like the COVID pandemic, is using molecular evolution to try to understand how viral genes and genomes are evolving. So, of course, viruses actually change their genomes and evolve very rapidly. Typically, viral genomes are copied by RNA polymerase instead of DNA polymerase, because a lot of viruses have RNA genomes. Um, and whereas DNA polymerase can proofread itself and is very high fidelity, RNA polymerase has no proofreading capabilities and actually makes a lot of mistakes. So we see viral genomes will accumulate, especially RNA viruses, will accumulate mutations very quickly. And that causes them to evolve very quickly. which is typically bad for the host, right? If we're talking about human viruses, that's bad for us. We see that with the flu virus, right? It's changing every year, and so you're having to get a flu shot every year. We've seen that happen with COVID-19, right? We see these new strains developing, and we worry about, okay, which ones are going to be more infectious or more severe? We have this COVID vaccine, but are the new strains going to be affected by that vaccine or not? So the bottom line is, if we have a deeper understanding of how molecular evolution occurs, right, we can apply this to the study of viral genes and viral genomes and maybe try to anticipate how viral genomes are changing, and that could help us produce better, more effective vaccines, make a vaccine that is going to anticipate some evolutionary changes and make it effective for a longer period of time. But that's just one example. There's all kinds of valuable uses for study of molecular evolution. All right, that is all I've got for you. Thanks.